Hi everyone, today I'm going to share my research project TRuC-T cell therapy with you. Before we get to it, let me first talk about the reason why I wanted to convey research on this. In 2018, there was an estimated 18 million new cancer cases diagnosed and 9.5 million cancer deaths around the globe. Indeed, the number are expected to rise about 50% by 2040. So I wonder if there's any breakthrough in the treatments for cancer other than the painstaking chemotherapies. And then I found this new technique, TRuC-T cell therapy, and began to research on this with my mentor, Dr. Boudreau. So TRuC-T cell's full name is T cell receptor fusion constructs. It effectively attaches anti-CD19 to T cell receptor complex, enabling them to recognize tumor surface antigens. And as shown in this picture, there are four essential parts in TRuC. There are EF1 alpha, anti-CD19, T cell subunits, and GFP. EF1 alpha acts as a transcriptional promoter to enhance TRuC protein production. Uh, the T2A in the graph is the cleavage site, where GFP is cleaved and detected as a sign of successful TRuC activation. To test TRuC assembly on individual TCR subunits, anti-CD19 was attached to each TCR subunit. There are five different kinds of TRuC. They are alpha 1, beta 1, gamma 1, delta 1, and epsilon TRuC. Changes in the arrangement of anti-CD19 can regulate the activity of T cell receptor, not only in surface expression but also in function. Of note, there are two single-chain variable fragments on the epsilon TRuC, while only one is fused to other units. So this maybe affect the efficacy of TRuCs, and it will be discussed in a moment. Yeah, to test drug efficacy in vitro, researchers used two approaches. One was by percent lysis of luciferase expressing NAM6 cells. The other was by an impedance-based assay using CD19 expressor HeLa cells to study the kinetic of cell lysis. Luciferase is a bioluminescent enzyme, which in this case serves as a reporter of successful lysis. In Figure 1, it is easy to see that there are stark differences in the lysis between vector control and two TCRs. Um, the vector control only lyses about 10%, while the most effective TCR, epsilon TRuC, exhibits about 65% tumor cell lysis. And in Figure 2, HeLa cells are tumor cells, which are represented by the y-axis. So after those T cells are added, the tumor cell index decreases quickly and finally decline to zero after 180 hours. Among all TRuCs, the most efficacious one was still epsilon TRuC cells, which is in accordance with the result in tumor cell lysis using luciferase. And to test the drug efficacy in vivo, they used a liquid tumor model. So researchers inoculate lymphoma and leukemia cells into the mice. After the tumor cells began to grow in mice, they injected them with TRuC cells and CAR T cells to treat them and test the efficacy of killing tumor cells. For epsilon TRuC, when about 100 million T cells were injected, the tumor volume kept close to zero, and no tumor could be detected at the end of study. Another advantage you can observe from this graph is that TRuC T cell uh, TRuC T cell have a very high survival rate for mice after injecting them, and this is very advantageous compared to CAR T cells because it has some cytotoxic effects of their therapy. So, in conclusion, TRuC-T cell is a generation of more effective CAR T cell therapy that use fusion constructs to incorporate anti-CD19 into functional T cell receptor. It can effective for vitro as well as vivo, and it has a lower